Hi, everyone. It's a pleasure to be here. And do I use this for the movement? Okay, great. Hi, everyone. My name is Hanadie Yousef. I'm the CEO and co-founder of Therapeutics, which we launched in 2018. And really, it's an honor and pleasure to be here today to tell you a little bit more about our company and our mission to ultimately mine systematically the secretomes of stem cells in order to identify novel stem cell secreted proteins with therapeutic potential, followed by translating them into engineered biologics that specifically target various mechanisms of aging to promote tissue restoration and improve function for the treatment of a variety of diseases, ranging from age-related through to rare. And we're doing this at Juvena through a robust, fully integrated, AI-enabled platform that leverages the latest in quantitative proteomics, multi-omics, and machine learning techniques in order to deconvolute these complex stem cell secretomes. At Juvena, we are completely obsessed with secreted proteins in particular. And why secreted proteins? Well, proteins that are secreted into blood circulation or hang out in the tissue microenvironments are in fact the natural biochemical messengers and drug-like molecules of our body that are responsible for modulating all sorts of processes, from immune response to activating our own body's progenitor cells to promote tissue differentiation and ultimately tissue degeneration. And given their natural ability to very specifically bind often multiple receptors to induce complex signal transduction cascades leading to the desired phenotypic response, they have a lot of great advantages which make them amenable to becoming blockbuster drugs. And in fact, for the last few decades, we've known this. I mean, think of secreted protein-based therapies like insulin or human growth hormone or epogen. But what's really interesting is even though we've known now for decades that secreted proteins have so much potential for us to really understand them and leverage them therapeutically, about 40% of the proteins, if, for example, in Juvena's proprietary library of stem cell secreted proteins have 0 to 50 citations, meaning they're either not studied at all or very understudied. And actually, an interesting fact is that about 30% of even known secreted proteins or predicted secreted proteins have no known binding partners, meaning we don't know what receptors they form or what complexes receptors they bind or what complexes they form. So there's really a lot of novel target biology that we can uncover at Juvena through our platform approach. And so the really underlying thesis is from looking at the causes of aging and tissue degeneration from the lens that during the aging process, proteins become in many ways dysregulated. Sometimes they garner mutations or issues in post-translational modifications. Sometimes they're under or overexpressed, but ultimately that leads to a loss of healthy signaling and what's known as homeostasis, which drives many degenerative diseases. And so at Juvena, we're ultimately mapping proteins secreted by stem cells to cells and tissues in which they can really target many of these mechanisms to help restore homeostasis. And our platform, really in this idea kind of around the approach and underlying platform that we've built is based on discoveries and observations made by myself, Professor Irina Conboy, and many others while I was in her lab that really showed that we can actually take proteins secreted by human embryonic stem cells, specifically isolating the soluble subfraction of secreted proteins. And this fraction itself was highly regenerative, not only in aged muscle, but in multiple human in vitro and then in vivo systems that we tested them in. And then at Juvena, while we have a platform that's sort of tissue agnostic, therapeutic area agnostic, we've really focused in on core organ systems that regulate metabolic health with the ultimate goal of wanting to restore all aspects of metabolic health. So while we start, what's kind of scary, as you can kind of see here in the image, is that when you're young, you really have this great balance between various tissues that modulate our metabolism, including muscle that's quite healthy. But when you get aged, you get extreme atrophy as well as an increase in adipose deposition and atrophy, not just of the muscle, but also bone. And so this can lead to sarcopenia, and you're really getting this, frailty, this muscle wasting. Unfortunately, you get this increase in inflammation. But that actually ends up signaling and really forming a feedback loop with, for example, fat in the immune system, where when you get that reduction in energy expenditure, insulin resistance, you start getting this accumulation of fat. There's actually more inflammation in that fat, and it really causes this negative feedback loop, which unfortunately, with every year passing for us and every decade, accelerates. And so really the novelty of our platform at Juvena is that we're creating this compounding database in a map of regenerative protein biology, where we take and identify proteins secreted by human pluripotent and multipotent stem cells, and then map them to receptors on various tissues and cells in which they can have specific disease-reversing phenotypes. And so when we really launched Juvena, one of the first things we did was establish our own proprietary library of proteins that are enriched specifically in the secretomes of human embryonic stem cells. And so the way we did that was by leveraging, first isolating the protein secreted by regenerative stem cell secretomes, such as human embryonic stem cells, compared to the same subfraction of protein secreted by either postmitotically, just postmitotic cells, differentiated cells, fibroblasts, or senescent cells, where those protein cocktails have differential effects when you add them onto aged or diseased tissues and cell models, meaning the regenerative secretome actually induces disease-reversing effects, while the non-regenerative either do nothing or are very modest or sometimes even quite pathological. And so we utilize the latest in quantitative proteomics and mass spec-based differential analysis to really hone in on specifically the proteins that are enriched in the more regenerative secretomes. And once we identify what those proteins are, we can actually start to add multiple labels, including things like their chemical properties, sequence information, evolutionary parameters, protein predicted or known protein-protein interactions that enables us to start building this kind of database that we can actually use to train models and start doing predictions of which proteins might be acting for which disease-reversing phenotypes. And so this first generation library that we built in 2018 and 2019, as shown here, really modulates various pathways and phenotypic responses that could have immense therapeutic potential, from the immune system to muscle development and pathways involved in insulin regulation, wound healing, and more. And what we also then continued to do once we started growing the company and secured our Series A financing in 2022 was to leverage then multiple stem and progenitor cell secretomes to really start building this database of proprietary library of proteins enriched in lineage-specific or pluripotent secretomes. And so our Juv2K library is what we've dubbed it. The second generation proprietary library is really even more expansive and really is characterized by all sorts of peptides to large proteins that are really diverse in the types of biological pathways that they're involved in. So there's just, we feel like it's like a gold mine of candidates that we can really start pulling out through this unbiased phenotype first screening approach we leverage. And so we've been able to actually leverage the library of about 2,000 proteins to map them to 140 different disease-modifying phenotypes across 17 cell types, and by today, link them to actually now 6 different organ systems that are all involved in overall metabolic health. And so the way that our systematic screening approach works is we start with our proprietary library of proteins enriched in these pluripotent stem cell secretomes. We then leverage a combination of bioinformatics and our deep learning models to rank order proteins in the library, which we predict might have a phenotypic response in a desired tissue organ system or model. Once we do that rank ordering, we screen, and once we generate hits, meaning actually see and pull out factors that are inducing the phenotypic response of interest, we can actually leverage that data and those features, what we call the true positives and negatives, to continue to build our database and then do iterative cycles of predictions and testing. So what's really exciting is it gets better as we go, and so as we continue to build the platform, we're becoming more and more efficient. Once we go from there, we move on to selecting some of the top hits and moving to extensive in vivo target validation, ranging in animals from the natural aged mouse model through to specific preclinical disease models. And once we've really generated ideally efficacy that matches what we think could have clinical potential for clinical endpoints, and ideally identify factors that are working in multiple models, we then move on to select the best ones and do our own protein engineering and lead optimization. And so we use a variety of data types and multi-omics analysis to really kind of leverage our ability to kind of rank, order, and predict what proteins might have efficacy, from looking at the disease pathophysiology of a target tissue or organ system or disease to ligand receptor mapping. We also really assess safety profiles. We make sure that these proteins, are enriched in our stem cell secretome, and we're always looking to see, does it actually change the protein itself or its targets in the pathway during aging? And so what's really also exciting about the platform approach is, while we start with an initial set of features, once we actually begin screening and generating data, we can leverage that to incorporate more information into our database and then leverage that to do better predictions as we go. So it's really compounding in nature. And once we really identify a hit, we do leverage, a variety of engineering techniques, some traditional, these days a lot AI assisted and enabled, to really improve these natural secreted proteins to make them more drug-like, such as improving the half-life. Ultimately, what we want are, many versions of, for like how, insulin is used for diabetes, subcutaneous injection, ideally weekly, month, or maybe even monthly based on the target product profile. And so at Juvena, we leverage multiple statistical and machine learning models to really accelerate our systematic screening and target validation and lead optimization through every step of the process, starting from the in silico candidate screening, where we're leveraging multi-omics disease data, CRISPR AI screens, genetic perturbation data, cellular phenotypic data, and clinical data. We move on to our high throughput screening and really the discovery approach, and leverage a variety of now different techniques there as well, including these sort of genetic perturbations is kind of some of the most exciting new stuff that we're doing there. And then ultimately as well in lead optimization, where we're doing things like, protein crystal structure predictions. We, of course, like many others, are taking advantage of the immense progress in the field and open access resources such as AlphaFold, and doing things to really predict and then improve and test these improved properties and engineering approaches that we can do to take these natural proteins and make them even better, more potent, and a drug for us to be able to use. And so at Juvena, in terms of, and I'm just going to be showing sort of examples of different kinds of techniques that we're using. And so we, for example, have recently established a genetic perturbation profiling platform to really ultimately continuously generate our own proprietary training data and leverage these CRISPR AI screens to generate more hits in a very rapid manner, and then also leveraging single-cell RNA-seq and transcriptomics to identify and understand those readouts and retrain our models. We are leveraging protein language models in order now to really, at the lead optimization stage, design novel sequence variants. So you can kind of see that here in terms of generating these structures, testing, and then really kind of going through the process and leveraging some of the latest techniques from diffusion models to ultimately some generative AI techniques as well. And not only are we sort of infusing different types of machine learning techniques into in silico, in vitro, in vivo screening and lead optimization, But as mentioned, we're really building our own proprietary internal compounding database where, we call this our kind of knowledge base, but this enables us to really take all this data, all this information that we're gathering, both the in-house multi-omics data generation, phenotypic readouts, image analysis, as well as a lot of publicly available data sets, and really integrate that into our ability to kind of internally visualize and rapidly understand what these hits are doing in order to validate them and select them as novel leads. And so this has led to Juvena's pipeline today, which, while we have managed to generate over 50 hits in 6 different organ systems, that has really fueled now a pipeline of biologics, starting with now our most advanced biologic, JUV-161, which is a fusion protein that enhances muscle regeneration and muscle metabolism, really targeting core mechanisms of muscle homeostasis, which we have decided that we're going to first develop it for the rare orphan disease of myotonic dystrophy type 1, which the FDA considers to be a progeria disease, an accelerated aging disease, and ultimately we want to leverage that to then expand to broader indications, including age-related muscle wasting and sarcopenia. We've also identified and have in preclinical development a second lead, dubbed JUV-112, which we're actually developing for obesity. And I'm actually going to give you a bit of snippets of both the data packages and what these 2 assets do to just share more, because it's really exciting. We do also have dozens of kind of early discovery programs, including doing things like combining our many hits that we've identified that can enhance muscle regeneration, prevent muscle atrophy, and improve muscle metabolism in combination with the GLP-1 and GIP agonists in order to really promote improved weight loss quality and prevent atrophy. And we've also generated hits across liver and lung tissue that can, even proteins with sort of dual functions, which can really inhibit fibrosis, but also then enhance specific types of regeneration, such as hepatic cells or the sort of AT2 progenitor cells and epithelium in the lung. And of those discovery programs, I've sort of emphasized them here. We have several leads that we've identified for that could be targeted potentially for idiopathic pulmonary fibrosis, including full in vivo data packages. We've identified factors that could be targeted towards NASH. We've identified multiple immunomodulators that have potential and potential therapeutic relevance. And we've identified factors that can really work for bone regeneration and actually for new cartilage formation. And so I'm going to zoom in and kind of show a little bit of data for JUV-161. And so the way that we discovered JUV-161 was really leveraging Juvena's platform as we built the foundations of the platform in order to map proteins secreted by human embryonic stem cells that can really drive aged or dystrophic human myogenesis. And so we started with our proprietary library of proteins enriched for therapeutic potential. We rank ordered the top about 100 that we felt could potentially drive aspects of muscle differentiation, myogenesis. We then screened those in vitro using various primary human in vitro models, taking primary human muscle precursors from either a variety of aged individuals or people with different types of dystrophies. And the goal here was to really identify factors that can promote better myogenesis and improve it to levels of really young, healthy cells. And so from there, we identified 13 hits that could do that. And as well as not just, many that worked quite well individually, but some that worked even better in combinations of, say, 2 to 3. And of that, we took the top 8 and validated them extensively in vivo. And what happened is we actually got 8 leads that worked in at least one preclinical model. And we tested a variety of preclinical muscle wasting models, ranging from our favorite, which is the natural aging mice. Once mice really get to that, kind of age of about 22 months, where you really start to, see sarcopenia start to happen and sort of enter that kind of stage of frailty, to injury in aged mice, where when the mice are injured, they get more fibrosis compared to if young mice are injured, and so we can, prevent that, to glucocorticoid-induced atrophy, to really mimic things like cachexia due to cancer or various treatments where you really get that inflammation-induced atrophy, to obesity, because unfortunately, as we kind of showed at the beginning, there is that crosstalk and communication between fat and muscle that really can accelerate the process of sarcopenia, and to even different models of dystrophies, including myotonic dystrophy type 1 and Duchenne's muscular dystrophy. And from there, we really got 3 leads in particular that showed robust efficacy in multiple of these preclinical models, and one in particular, which is now our lead on its way to the clinic later this summer, that we nominated, and I'm going to share a little bit more data about that. And as noted, we've really then continued to expand the program to what we're now calling the muscle catabolism program, where now that we've expanded the library to over 2,000 proteins, we're screening and actually generating many more hits that are specifically targeting aspects of muscle atrophy and muscle insulin regulation, metabolism, and it's a really exciting project that's really advancing. So JUV-161 is an engineered signaling fusion protein derived from the human embryonic stem cell secreted protein and family member that we all heard of, insulin-like growth factor 2. So IGF-2 was discovered through our RJUV1K library screening, and it specifically signals to drive muscle differentiation, promote survival and maturation of new fibers, as well as targeting aspects of metabolism. We have introduced certain mutations that improved its stability and fused it to a human serum albumin, where its half-life went from the natural half-life of around 20 minutes to what we believe is going to be over a week in people, based on our humanized mice, dog, and rat studies. And so 161, so IGF-2 really signals through 3 receptors. What makes it unique from the IGF-1 is a molecule is it specifically also targets IGF-2 receptor, which is important not just for lysosomal trafficking, but uniquely induces sphingosine signaling, and it activates AKT signaling in order to ultimately promote this sort of muscle metabolism and muscle regeneration. And so, for example, when we take mice that we establish at Juvna, a DM1 mouse model, we call it myotonic dystrophy type 1, where this mouse model is doxycycline inducible in the sense that when we feed the mice doxycycline, a promoter turns on the gene mutation that causes the disease, which is a DMP kinase gene that has multiple CTG repeats that then causes an RNA hairpin loop to express in multiple tissues. And over time, the mice will start exhibiting muscle wasting, frailty, and multi-organ degeneration, as well as metabolic dysregulation, just like people living with the disease. And so I'm only showing kind of snippets here, but what we can see is not only an improvement of specifically muscle metabolism and the increase of glycolytic fibers, which are reduced in aging and in dystrophies. We see a reduction in blood glucose and better glucose regulation, which is really important for both natural aging, but as well as people with this disease. And not only are we preventing that steady decline in strength, as you can see on the right, but we're actually, even in the context of the genetic mutation still there, not just preventing, but actually improving strength and helping to restore muscle function, which is really, really important. What's exciting about 161 is it works in a variety of different models that can really mean it can translate in many ways to become a pipeline and a product, where we want to show POC in this rare orphan indication, this disease population with very select inclusion-exclusion criteria and ability to get clinical endpoints in the very near future, with our actually phase 1 starting this September, assuming the regulatory filing we're in the midst of goes well, and then phase 2 starting next year. But then we're going to leverage that to really expand to the diseases of aging that really afflict, ultimately, eventually all of us, meaning age-related muscle wasting sarcopenia, potentially sarcopenia associated with obesity. For example, it could potentially be applicable for folks with injuries, and really just various myopathy. So we're really excited about this insulin of muscle that we're developing that will be subcutaneously administered weekly or bi-monthly. As we continue to expand the platform, after really kind of focusing and mapping proteins to muscle, we began looking at other metabolic tissues, including adipose. So beginning in 2021, we began really screening the second generation library to identify factors that can specifically enhance lipid metabolism and really target adipose cells directly. And that's where our second now nominated lead came from. And so what's interesting with JUV-112, and I won't be able to share the name of this protein, but what I will say is it's incredibly novel. It was not known to be secreted. We don't know any other kind of company as of now or academic lab in the world really studying this factor or these targets. And the way that it acts is it acts directly on adipose cells to enhance lipolysis. And as you can see here, in primary human adipocytes, we're able to, on a dose-dependent manner, reduce the size of the lipid droplets and really, on the right, improve the activity of lipolysis and triglyceride ultimately breakdown. And so jumping forward into the DIO high-fat fed obese model, even in this first unengineered form where there's actually a lot of room for growth here, we are inducing about 11% body weight loss, about 15% specifically fat mass loss, and importantly, we're doing it, as you can see on the right, without changing muscle mass or parameters of strength. Meaning, compared to the most potent incretin, which does unfortunately, cause not only muscle mass loss, but the loss of strength, as shown here by grip strength, we really don't affect that, because what's really exciting is our mice are still eating, right? This is not a starvation-induced weight loss mechanism. This is a mechanism where, in many ways, you can kind of still eat your burger, but you're just getting improved lipid metabolism, so you don't have to feel guilty after. And you can see that on the right, where our mice are eating just as much as the control, where of course with the positive, the incretins are doing what we expect. They're really causing starvation. And what's even more exciting is, we're not getting, of course, so far in this form, as severe a kind of, fat loss, weight loss as the incretins, but we're getting just as good metabolic rejuvenation. Meaning we see, as you can see on the left, improvements in glucose tolerance and insulin sensitivity, really just 2 levels of the most potent, GLP-1 agonist out there. And what's exciting is that one of the, what our drug does is it's actually first targeting organs such as the liver before the kind of subcutaneous fat to really reduce the concentration of sort of those an amount of that lipid. So in liver, for example, we really see dramatic attenuation of not just the fat, but also fibrosis, as seen here on the right, and you can really just see that just from it's so obvious from the pictures alone before we even do the quantitations. It's when we dissect the mice, and it's really impressive. With that, I know I kind of went through a rapid amount of information and data, but I do want to take a moment to really thank. I'm just the messenger at this point. I really want to thank my team. Specifically, my co-founder and chief scientific officer, Jeremy O'Connell, who really I met at Stanford in 2017 after he had completed 5-year postdoctoral training in the lab of Steve Gigi at Harvard, and his specialty is quantitative proteomics, machine learning. And then importantly, one of our core founding employees who, it was Jeremy, and then 1 year later, Tak joined, and we really kind of, built the platform together. And so Tak is our director of discovery and platform. He had spent 6 years in Helen Blau's lab at Stanford before joining Juvena. And I highly recommend folks consider inviting him to talks. I do not do our AI technologies and techniques justice compared to how Tak describes it, given he's really been incredibly innovative in leveraging the latest AI techniques such as neural networks, diffusion models, and the latest in generative AI to really innovate both in silico, in vitro, in vivo, and at protein engineering. And with that, I'd like to thank you for taking the time to hear my presentation, and I'd be delighted to answer questions.
Thank you so much, Hanadie. So yes, please, questions for...
This is probably very naive, but do you work at all in influencing post-translational modification to innately generate the proteins or some other epigenetic changes so that you are endogenously creating the proteins? That would be kind of a step back from squirting the proteins indirectly. Very nice presentation, by the way.
Thank you so much. I'm not sure exactly if I understood the question, but I will say that it's really important that we kind of ensure that the recombinant proteins we're developing do have the same types of post-translational modifications, methylations as what naturally occurs from, when the stem cells produce and secrete them. So we're doing our best to, for example, use mammalian cell lines in producing them, and we do the, the proteomics to basically assess and understand and really take into account if we've changed those modifications. But we're really going after we're taking advantage of sort of the modality that has been the most lucrative, the most successful, in the history of kind of drugs to date, which is recombinant DNA technology and the ability, once we know the sequence, to produce it externally, mass purify it and distribute it to millions, if not billions of people. So we really wanted to try with a proven modality first.
Yeah, so I think Walter's question was basically, are you considering delivering your proteins as mRNA or with gene therapy?
Exactly, and so that's what I actually figured at the end. And then so we're not going to, for now, we want to deliver it as as biologics, because we feel just like the way that insulin has been so successful as a pre-filled syringe, daily administered therapeutic that has saved hundreds of millions of lives for people with the metabolic disease of diabetes, we think that could be a good first approach in adoption. But we're very open as we evolve to considering things like mRNA deliveries to be more tissue specific, although you'll lose the ability to really control dosing, which is important because we want to maintain homeostasis and physiological levels, not saturate a system. So there's also concerns with that.
Yeah, thanks. In the end, you mentioned fibrosis, which is associated with sarcopenia. How about cellular senescence or inflammation more generally? Do you also find that you are tackling this, that your intervention influences, especially senescence?
That's a great question. We've actually not... I know we're really interested in seeing if we're kind of reversing, and making senescent cells active and sort of rejuvenating them in a kind of reprogramming fashion. I will say that, many of the factors in our stem cell secretomes are inducing, the pathways involved with sort of partial reprogramming, but on an individual sort of, protein level, when we do those injections, we haven't yet kind of looked too deeply into that, but that would be a great kind of avenue and an observation we should probably do.
All right, thank you. Thank you so much.